Welcome. First and foremost, I want to thank you for choosing to watch this video today on the applications of antibody technology. We're going to dive in and take a look at how scientists have been able to alter and utilize antibodies for purposes other than fighting diseases. Within this video, we'll look at 3 specific applications, and then afterwards, you're going to be conducting a lab simulation of directed evolution of a B cell to produce a more specific and more useful antibody to fight a specific disease. Let's move on. As always, we begin with our student learning objectives. You should be able to understand how antibodies can be used in a variety of technology applications, and also be able to explain the concept of directed evolution and how it can be used to increase the efficiency of an antibody. So let's talk briefly a little bit about some forensic applications of this. Much like matching DNA samples at crime scenes, antibodies can be used in a very similar way. Each individual person makes their own unique antibodies. Even if they're for fighting the same disease and in 2 separate people, they're still different and still unique. Each individual's antibodies are unique just like DNA and therefore can be used to match samples. Now, what makes this different and more useful than, say, a DNA analysis is that these are actually significantly cheaper and require less time to run the required test. So not only is this a way that you can match evidence at a crime scene with a potential suspect, but it's also more efficient and cheaper to run. We also have the ability to test for antibodies using a technique called ELISA, or enzyme-linked immunosorbent assay. It's a way to determine if antibodies are present inside of an organism, something that is very useful to determine if an individual has already had an illness. In order to run this, a blood sample is taken and then it is reacted with an antigen, while the sample is altered to have special protein markers on them. If the antibody bonds, then the protein reacts with the substrate and produces a colored product, thus representing a positive result. This is, again, very useful antibody technology and forensic technology to be able to determine if an individual has had a particular disease or illness and has the antibodies that are able to fight it off. Lastly, we're going to look at the directed evolution of antibodies. Now remember, when we talk about evolution, those with traits best suited for the environment are more likely to survive, reproduce, and pass those traits on to their offspring. Well, in lab, we can actually mimic this process of natural selection to get a desired outcome of an antibody. Think about artificial selection, which is picking and choosing what types of traits we want in a particular organism. Dog breeding, for example, is artificial selection. We're going to use random genetic mutations of a B cell genetic code to produce antibodies to work differently. When we change the genetic code, we change the protein through the process of creating mutations in the genetic code. When the mutations are made in the genetic code, the protein or antibody that is produced is going to be different than the one that the B cell would normally produce. We then take a look and isolate and select B cells that function more efficiently. And then we repeat this process over and over again until we get a desired outcome. We make lots of copies of them and repeat the process. If we look at the first generation here, and this is an oversimplified version of it, but still, we want the very end of the antibody to be able to create as many intermolecular forces as possible to be able to bind to the antigen. Well, if we look here in this first generation, there's been several mutations. We're going to take the 2 that seem to work most effectively in binding, and then we repeat the process. And then we repeat the process again until we get an antibody that gives us our desired outcome. There's going to be a very good simulation of this later on in the unit. Once again, hopefully you have your student learning objectives and you're good to go. Thank you so much for watching and have a great day. Bye-bye.